After we have collected all of our data from our experiment, the last step is to analyze that data and decide whether or not that data can be used to support or reject our hypothesis. So our data will generally come in one of two forms. And this data is going to be recorded. You're going to make a nice little chart that's going to display all of the information that you have collected. Now, depending on how you're presenting or publishing this, you might select only some of the data, you might select all of the data. It really just depends what information you are trying to convey. But we're either going to collect qualitative data, which are descriptions, or we're going to collect quantitative data, which are numbers and value. And the type of data that you collect really just depends on the goals of your experiment and what you need to support your hypothesis. So qualitative data are absolutely important, but they can be subjective, meaning that if you look at a quality like large or small, my idea of small in a particular situation might be different than your idea of small in a particular situation. And so without a sort of set range for certain values, it can be really hard to determine where your data is going to fall and therefore lower your ability to use that data to support your hypothesis. When we're designing an experiment, we really want to try to get those quantitative numbers and values. And that's because it's really easy to set a range. It's really easy to set a maximum and minimum value. And along with that, when we have numbers and values, we can use statistics to help us determine whether or not our values are significant. Meaning we can use statistics to tell us if we have a change that we can indeed call a change from our normal or our control group, and that we can use in order to support or reject our hypothesis. So as we're thinking about designing our experiments, we really want to try to get measurable values. Now, what you do with that data depends on the type of data you have and your goals for the experiment. So very often we'll do statistics on our data. Usually we want to repeat our experiment with as many different sample groups as possible, a large sample size if possible, because that's going to better allow us to see trends in our data. It's going to give us multiple data points to know that we can be confident that our data is accurate. And it's also going to allow us to see outliers, or those data points that might not really make sense and sort of be erroneous data or one-off data points. So when we repeat our data, when we have a large sample size, we're going to get more credible data and we're going to get more credible statistics. So we might calculate something like the average value, and we can do lots of different statistics. In our course, we're really only going to do one statistical test. This is called the chi-squared test, and we'll do it in our genetics unit. But you might be asked to calculate an average or a mean on some of your data. Most of the math that we're going to do in this course is pretty simple. Once you have your data and you've done your analysis on it, you usually want to display that data in some way. Now again, the way that you display it really depends on the type of data that you have. But very often in this class, you're going to be asked to make a graph. The important thing about any graph that you make is that you label the X and the Y axis correctly. So on this graph on the right, I have a line graph that shows changes in wolf population and moose population over a long period of time. And on the bottom, we have our time. On the sides, we have population size. Now, for every graph you make, your x-axis is always going to represent the independent variable. And the y-axis is always going to represent the dependent variable. So as you're designing your experiment, you want to keep in mind how you might collect your data and present your values. So we'll talk about graphs a little bit more detail in some of our labs, but the key to making an accurate graph is always labeling those x and y variables correctly. So we might use a line graph to show changes or trends over time. Line graphs are great, especially over long periods of time. If you have a lot of data points over long periods of time, if you have a lot of different categories, line graphs are a really great way to see trends and to see changes. We might also use a bar graph in this class. Bar graphs are really great for comparisons. So in this bar graph, we have average height of big land animals. So we have 4 different types of animals, and we have males versus females. And so we've got a lot of densely packed information in this graph here. We're very easily able to see the differences between 4 different animals and the differences between males and females within those animals and among those animals. So we usually are going to use those bar graphs when we're really trying to see comparisons. We want to try to stick to the line graph for time and the bar graph for comparisons when we're not looking at changes over time. Another example we might see is a pie chart. This one shows the percentage of time that cells stay in each part of the cell cycle. And again, this is another one that we might use for comparison. Sometimes there are different ways to show our data, and that's perfectly fine. Usually for our labs, I'm going to tell you what kind of graph we want to make, and we'll get some practice with some different ones. So we're going to use this data to help us draw our conclusions. So as I said, sometimes we're going to use statistics. We won't use statistics a lot in this course, but you might see some statistics in our data sets that come in our textbook or that come in class examples. And I'll explain those statistical analyses as they are appropriate. But as you move on in science and you start analyzing your own data sets, you'll want to think about what kind of statistics you might need. Of course, you'll need to take statistics, but that's part of learning how to analyze your data. The important thing that we get from that statistical analysis is whether or not our differences are statistically significant. And when you have a statistically significant data set, this means that you have registered a change between your control group and your experimental group that you can use to support that hypothesis. The alternative is that maybe you don't see a statistically significant difference, in which case you would use that to reject your hypothesis. So when you are writing out your conclusions, you always want to make that statement. I supported my hypothesis that. And then you're going to restate it, whatever that hypothesis was. So you want to make a very clear statement whether you have supported or rejected your hypothesis. Now, it's also appropriate to talk about some other things in your conclusion. And for our class, in our labs, we'll ask you to do certain things. Sometimes we'll just ask you for a statement. Did you support or reject your hypothesis? Other times we're going to ask for a little bit more analysis and application. And all of these questions are appropriate for your data analysis. So what did you learn? Did you find anything interesting or significant in your experiment? Are there other studies that can be done now that you have supported or rejected your hypothesis? Does that change your question? Do you need to change your hypothesis? Does this lead you to another hypothesis or further investigation? Were there any experimental problems or limitations? This one is really important. This is where we want to talk about those uncontrolled variables. Because remember, those uncontrolled variables reduce your confidence. Even if you support your hypothesis. So just because you have supported your hypothesis doesn't mean that there were not problems and limitations, and you always need to address those limitations and those uncontrolled variables. And then, of course, you want to always talk about your data in your conclusions. Why were your study and your data significant? Why were you able to use your data to support your hypothesis? You want to address specific data in your conclusion because you don't want to just present the data and then make a statement. You want to make sure that the reader understands the connection between your data and your conclusions.